All right, for this embryonic development video, learning target tracker video, the first I can statement is I can define cell differentiation. Cell differentiation is very similar to like a specialization when you think about doctors. So when doctors go to med school, they come out, they're a doctor. Then they go into a residency and they specialize. And so instead of just doing all doctor stuff, now they do just OBGYN or just surgery or just neurology or whatever. So they're basically narrowing their focus and focusing on one field and learning a lot about that one field. Differentiation is kind of the cells version of that. Basically, cells are, when they first start out, like stem cells, so these are, let's say these are two identical stem cells, sister cells, identical, can't spell them right at the same time, apparently, and they have all of their genes on, okay? And then they go through this process of differentiation or specialization. Differentiation. And what that means is they start turning certain genes off. And so now, let's say they turn this one off, and this one off, and this one off. And so now the genes that are left on make them a stomach cell. Okay, so now they can't do everything like they used to be able to do. Why do I not have this? Sorry, y'all. There you go. So they're identical. All genes are on. They go through differentiation. This one gets a different signal, so it turns different genes off. It might still turn that one off, but now maybe it's turning off those. Okay, and it becomes, we'll go with a muscle cell. I was going to say a neuron, but that does not look like a neuron. And so now they have become two different, I know stomach cells can be muscles too, but let's say this is a cardiac muscle. Okay, we'll say that so that we'll know for sure it's different. So cardiac muscle cell. So because they went through this process of differentiation, they turned different genes off. It allowed them to focus in on just the jobs that they needed to do and make just the proteins that they needed to make for those jobs. And that's how they became different. And that's why it's called differentiation. So that's what cell differentiation is. The process of induction is one of the ways this happens. So differentiation can happen by a couple of different ways, but induction is one of those. And induction is simply where one cell tells a nearby cell what to do. So this one says, the signaling cell says, hey, I'm becoming a stomach cell. Since you're right next to me, why don't you be a stomach cell too? And so it sends these signals out that say stomach cell, and they bind to the receptor, and it sends the signal. So this is reception, transduction, response. And the response is to turn off all the genes that don't make it a stomach cell. And so now the only genes that are left on are stomach cell genes. And so this cell right here will become a stomach cell as well. So induction is kind of like peer pressure, kind of, hey, why don't you be what I'm doing? Why don't you do what I'm doing, or you be what I am? I can discuss the events that happen in fertilization. So we talked about fertilization. Basically what happens, I'm not even going to try to draw this. Basically what happens is the sperm, there's millions of sperm come into this egg. And this egg's like, whoa, 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 I just need one of you. And so the sperm has enzymes in its head that are released that bore through this jelly coat or digest this jelly coat. Once it gets through the jelly coat, that opens up the receptors, and so the sperm can now bind to the receptors. This makes sure that it's the right species. And this is really important in external fertilization because there's a lot of sperm and eggs floating around in the ocean. Gross thought, I know. But you want to make sure that the right sperm is fertilizing your egg. And so the sperm receptors bind to the egg, and the egg says, yep, you're the right species. Your proteins fit my receptors. It lets the sperm in, so the nucleus comes in. The rest of the sperm is kind of left out and degraded. But the nucleus comes in, and it's going to fuse with the egg's nucleus. But meanwhile, after it lets sperm number one in, it throws up a force field that prevents other sperm from coming in. And we call that the block to polyspermy. We don't want polyspermy. We want one sperm getting in that egg. And so the eggs have to do something to prevent any others from getting in. And so that's called the block to polyspermy. After hours or so, I think in sea urchins it's not even that long. It's like 20 minutes. The nuclei fuse, and once the nuclei fuse, then the egg will start. I guess it's now a zygote. Will start dividing, start going through mitosis. I can explain pattern formation. All right, once that zygote is formed and it's early in development, it's going to go through a process called pattern formation. And this is simply the setting up of the body. It's basically saying this end, these cells on this end are going to become the head. These cells on this end are going to, so head. These cells on this end are going to become the tail. This is going to be the top, this is going to be the bottom, left and right, and those kinds of things. And this is going to help the body put the organs and things in the right place by setting up those axes first. All right, then I can discuss the role of homeotic genes. Homeotic genes, or you might hear them called Hox genes, are super important. When we see large-scale birth defects, it is generally because the homeotic genes have been impacted, because they are setting up the body in big things like, oh, you have 2 arms and they're coming out of your shoulders. You have 2 legs and they're coming down from your hips. Those huge placements of big things like that are due to homeotic genes. And so they are what we call master control genes, and they are the reason we have different body forms. So when you look at a fish, it doesn't look anything like an elephant, right? Because their Hox genes are different. They have different mutations. There are some that are the same. They have Hox genes that say, hey, we need a brain. Hey, we need 2 eyes. Hey, we need 4 legs. Okay, so some of their Hox genes are the same, but there are others that are different, and that's why you get different body forms. I was thinking a mouse and an elephant. Fish do not have legs. But anyway, sorry, in my mind, I was picturing a mouse, not a fish. So anyway, with a mouse and an elephant, they have the same ones that say 4 legs. A fish and an elephant would just have the same ones that say brain and 2 eyes. Okay. I can discuss cleavage. Cleavage is simply a term that means rapid mitosis or rapid cell division. So after it's fertilized and it forms that zygote, then it starts to divide. So 2, 4, 8, 16, 32, 64, 128, so on and so forth. And that rapid mitosis or that rapid cell division is known as cleavage, because to cleave means to cut. So you're basically cutting these cells in half over and over and over again. I can discuss gastrulation. Gastrulation literally means formation of the stomach. And that is a part of gastrulation, but it is just a small part. Gastrulation is where we have this buckling in. I feel like I have a better picture. Here we go. This buckling in of the, so here's your blastula, which is what we have at the end of cleavage. So we go from zygote to morula. It becomes a hollow cell called the blastula. It starts to buckle in. That process is called invagination, and that's going to form our gut. And so what we see, it's called the archenteron. That space that's being formed as we push this all the way to the other end is called the archenteron. It's going to become your digestive tract. And so that's why it runs all the way through you, because you basically have a hole that runs through you, or a tube that runs through you, with one end to be in the mouth and the other end to be in your anus. And so this is forming that tube. So that's what gastrulation is, and that's why it's called that, because it is forming your stomach way down the road. But also during this process, during that formation of the archenteron, the outside of the blastocele, so the parts around that tube that's forming, are going to organize themselves into 3 layers: the endoderm, mesoderm, and ectoderm. And those are going to give rise to different tissues in your body. So that also happens in gastrulation, and that's huge. So we talk a lot about the archenteron being formed, and that gastrulation, gast means stomach. But like I said, that's just a small part. The formation of those 3 layers, those 3 derm layers, or what we call germ layers, is the most important part of gastrulation. All right, and this is showing that that tube going all the way through. So you have that digestive tract. So like I said, you are basically a big donut or a big tire, because you have a tube that runs through your entire body. And then your body is kind of wrapped around that tube. It's kind of an interesting way to think about your body. But all right, I can discuss organogenesis. We don't talk much about that. We don't go into a lot of detail, I should say. Organogenesis, genesis means creation, so it is literally the creation of organs. Once those 3 layers are formed, and here they're shown in kind of a blue, and I thought it was labeled in one of these, but maybe not. But the blue, the red, and the yellow, those are the blue, the red, and the light orange, whatever that color is. Those are going to be your 3 layers that are going to give rise to all of your organs. So once that happens in gastrulation, then you can start forming the heart and the stomach and the brain and all that kind of stuff. And so that's a big part of embryonic development, but it's going to happen later. All of this is happening super early. So I'm not talking about like at 9 months you're forming your organs. No. Your organs are formed really early, and that's why you've got to be really careful the first month or 2 that you're pregnant. because that's when all this stuff is forming. The first couple weeks, actually. And then the last is I can explain the role of gene expression and regulation in the development and differentiation of cells that occurs during development. So we already talked about how gene expression is super important in differentiation, because these white open genes are being expressed and the ones that are shaded are no longer being expressed. So we've regulated that. We've turned those 3 off and left those 3 on. So that's regulation. That's a choice we're making. And that's what caused these cells to become different. And so gene regulation is super important because it turns off the genes that we don't need. Okay. As far as why this matters in embryonic development, every one of these steps, when to do mitosis during cleavage. Okay, so when do I go from 2 cells to 4, from 4 to 8, from 8 to 16? When do I start pushing out and going from being a morula to a blastula? When do I start that invagination process where it starts to buckle in and form that archenteron? How does that happen? There are proteins that are making all of these decisions for the cell, and those proteins are being expressed from the DNA. And so it is imperative during embryonic development that gene regulation is very well controlled, so that you turn genes on at the right time and you turn them off at the right time. You don't want to leave them on too long. You don't want to turn them off too early. You don't want to turn them on too early. So all of these genes have to be turned on at just the right time, left on for just the right amount of time, and then turned off at the right time to ensure that the baby, the puppy, the kitten, the joey, which is what we call baby kangaroos, all of those things develop just as they should. And so it is super important when it goes well, when gene expression and gene regulation of expression occur just as they should, you get a healthy, happy baby. And so that's kind of the nutshell of how gene expression and regulation controls differentiation and development, because the proteins run the show. And so the proteins tell everything when to happen. And so if the proteins don't say it, it doesn't happen. And so you want to make sure you have those proteins produced, gene expression, at the right time. And that's it. So if you have any questions about embryonic development or anything that you're still unsure of, let me know and we'll go over it. I hope that helps.